Hi, my name is Sarah Goodwin, and we're here today with Dr. David Morgan, who is a professor at the University of California, San Francisco, and Dave is an avid book collector. So thank you for joining us.
My pleasure.
And we'll start with, how did you begin collecting books?
It started many moons ago. I was doing it when I was a kid, actually. And I used to go to the used bookstores in my hometown all the time, even when I was in my early teens, and I was fairly obsessed with them right from the early days. And so I've always been interested in books. The last 15 or 20 years is when I've really gotten going on the important old stuff that I have now.
How big is your current collection?
I'm not at liberty to say. My wife thinks it's way too big. That's all I can say. It's huge. I have hundreds of things. I have boxes in most closets. I have shelves in most rooms in the house. And so it's a bit out of control. And in fact, in the last few years, I've really tried to get a little more focused and just get a few good things instead of filling things up with a lot of bad things. And so, but it is, I don't know, hundreds, thousands maybe.
What types of books did you collect when you were younger?
Well, believe it or not, my dad was an airplane mechanic, and so I was heavily into airplanes. And so I loved books about World War II airplanes when I was a teenager. So I have stacks of those in my basement that I still have. But then as the years went by, I became interested in a lot of other things as well.
What was your first big find?
Oh, well, I mean, there was this transition period back in the mid-90s when I became serious and realized I could get the really old, interesting, original things from hundreds of years ago. And so, in a way, those early books that I got at that stage were kind of the highlight in a way, because it was all new and exciting, and I thought it was really fun to have a book that was 400 or 300 years old. Although in retrospect, what I was buying back then was really not very good stuff. I didn't know what I was doing in those early days, and so it takes many years actually to learn what to look for when looking for a book, and collecting condition, the binding, all these other issues that you have to think about.
So let's talk about some of the books that you brought today.
Yeah, I thought I'd bring along a few highlights of one of the main interests of what I do, which is collecting books in the history of cell division, and basically going back to the 1700s or so, when cells were first witnessed through microscopes and other little optical instruments, and then working up into the 1800s when the cell theory came along, and when people first started realizing that cells reproduce by division. And so there are dozens of great scientists involved in this process of making their way from the 1700s through to the roughly late 1800s when all these things were found. And I can't possibly, I have a lot of their work at home, but I couldn't bring them all in. But so what I did was I brought a few of the highlights, the ones that kind of turned me on the most. And in a way, a lot of people like to start with Leeuwenhoek, the Dutch microscopist who first saw little animals under the microscope. And he saw a lot of different organisms through his microscope, and little animals, he called them, as I said. And in some cases, he may have been witnessing cell division, but he didn't really know what he was looking at. And in fact, it's unlikely that he ever actually witnessed or reported the division of a cell, of a single-celled animal. And it wasn't until a little later, so he was in the late 1600s, early 1700s. It wasn't until more like the middle of the 1700s that people actually started describing the division of single-celled organisms. And the first guy to do that probably was this fellow from Geneva called Abraham Tremblay. And he was a really interesting character who grew up in Geneva, and then in his 20s moved to Holland to become a tutor for the children of this aristocrat who lived on this giant estate in Holland. And on that estate, Tremblay basically taught these two kids, while at the same time exploring the grounds of the estate and exploring the water of those grounds, and finding little animals that lived in there, and then looking at them under a magnifying glass, essentially. One of his biggest discoveries was that he found this little organism that we now call Hydra, which had been described previously, but not to the extent that he did. And in studying Hydra, he was really curious about whether Hydra was a plant or an animal, because it looked a little like both. And in one of his experiments, when he wanted to figure out if it was a plant or an animal, he decided, well, maybe if he cut it in two, an animal would die, probably, whereas a plant might continue to grow, or might at least survive in some way. But he was absolutely shocked because when he cut Hydra into two parts, the two halves regenerated to form two whole organisms, which then moved around like animals. And so it became clear that Hydra was an animal that could be cut in half and regenerated. And that was a mind-boggling result at the time. And when word started spreading, he communicated with some of the great scientists of the day through letters, of course, and when word started spreading of this discovery, it just caught on like wildfire. It was like one of the biggest things of the century. It's sort of like the structure of DNA is to the 20th century. Regeneration of Hydra was a huge, big deal. And so people in Britain, people in France, all over the place were repeating his experiments to make sure they were true. And then eventually, he published this work in a journal called the Philosophical Transactions of the Royal Society, where he was actually invited to submit the work because everyone was so excited, and they actually dedicated an entire issue to this one topic. So he got the cover, essentially, and I happen to have a copy of that special issue of the Transactions in which his work was first published. And so it's mostly words, of course, but there are some nice illustrations, like this one here of a Hydra bound, stuck onto a twig of some sort. And then in this paper, which is written in English, by the way, so it's actually totally readable in this wonderful 18th century scientific English. It was translated from French by someone at the Royal Society, and the English is just a lot of fun to read, and it really captures every moment of the discovery. Essentially, every little thing is described in wonderful detail, and right up to the point where he talks about cutting it in half and observing over a period of days how these two new Hydra would regenerate from the two cut halves. And so, and then this article is accompanied by commentary from various other people, including at the front, there's a letter from the aristocrat, Count Bentinck was his name, whose estate he worked on. That's here at the front end. And then following the article are commentaries from other famous scientists. And so the whole thing was a really major discovery in the 18th century and created quite a stir. And then, so this came out in about 1743, this article. He was immediately made a fellow of the Royal Society because of this, and he won their Copley Medal, which is their major award for science every year. And then in the year after that, he published this incredible book that came out about a year after the article in the Philosophical Transactions, in which he summarized everything he had done with Hydra over a period of 2 or 3 or 4 years. And so this book contains all kinds of wonderful observations in French of every little experiment, every observation he made about Hydra, beginning with the basic description of the organism all the way through to these incredible experiments in which he cut it in pieces and analyzed regeneration and analyzed all kinds of other features of its behavior. But this book is not just great for its science, but also because it's one of the most beautiful scientific books ever. Certainly in the 18th century, it's considered one of the classics because it contains so many beautiful illustrations as well. Because he recruited a friend of his to do these copper engravings of the Hydra, and those copper engravings are now considered to be among the most gorgeous scientific illustrations ever done. And so you can see all these really delicate little Hydra diagrams that were drawn on copper plates, of course. These are copper engravings, and so this artist by the name of Lyonnais was responsible for producing these engravings. And so this book is just filled with gorgeous engravings of Hydra in various states. It also includes illustrations of the famous experiment where he cut the Hydra in pieces. Way back here, there's a figure showing the two halves of a Hydra that's been cut, and then following the tail half as it begins to regenerate a head, essentially. So, and in fact, this figure has all kinds of fun other experiments where he actually turned Hydra inside out by inverting the inner cells to come out onto the outer cells and then observed what happened to those cells. So he was doing lots of really interesting and innovative and creative experiments. And what's really fun about the book is it also includes illustrations of him at work, including this really classic engraving here showing Tremblay at work with the two students on the other side of the table, and with little jars of Hydra all sitting on the windowsill behind him, and the grounds of the estate in the window outside. So it's really captures this amazing moment in scientific history when this guy was doing all these experiments essentially all by himself. He communicated a lot by letters with some of the famous scientists of the day, as I said, but he was basically working on his own and just having a great time for years studying these organisms. And so in a way, this book, part of the appeal of this book, in fact, is that it is sort of a relic of a different era in science when it was completely unfettered by all the modern issues that have sort of taken over in science, issues of funding and institutions and careerism and all the other issues that now influence how science is done. This guy was doing pure, unfettered science, and so this is a great relic from that era. So Tremblay, but the amazing thing about him was that this wasn't all he did. He actually also looked at a lot of other little animals in the ponds of the estate, and that's where we get to cell division. So this was a bit of a sidetrack from cell division, but it's the foundation for what came next, which is that he started looking at protozoans as well, that he also called little animals, essentially, that were essentially little single-celled ciliates like Vorticella and Stentor and some of the other large single-celled protozoans that are big enough that you can actually see them through a magnifying glass. Because he didn't actually use microscopes, he used really fancy, high-powered magnifying glasses. And so when he started looking at protozoans, he witnessed them dividing. And his descriptions of that protozoan division were published a couple years later, also in the Philosophical Transactions. And so I have copies of those as well, of papers in which there are basically two papers, one from 1744 and one from 1747, in which Tremblay describes the reproduction. He describes these little animals and then describes how they divide. And he actually illustrates the division of some of them. So for example, these communal vorticellids, like this guy right here, that which are pretty well understood organisms at this point. They have this little bell-shaped head on them with a long tail, and they divide by longitudinally dividing down the middle, essentially. And then they form into these communal multicellular gatherings, essentially. In a way, these two papers here are really the first description anywhere in detail of cell division of any sort. And so these are kind of the foundation stone for studies of cell division. Although ironically, as you know, he did not know he was looking at cells, because this was 1740s. And at that point, cells were considered something that were probably existing in a lot of plants. Their importance as the fundamental building block of all organisms was not even close to appreciated. So it's an interesting case of someone looking at something and describing it in detail, but not knowing, not having any idea really of what he was seeing, which I always like to think about as a lesson for today as well, because a lot of us in the lab are busily describing things in detail and not really understanding them in a way that we will possibly be able to understand, hundreds of years from now. After Tremblay, the next real wave of cell division work came from various people, mostly working in the early 1800s. Another really fun paper came along in 1824 from a pair of French scientists called Prévost and Dumas, who were the first to describe the cleavage of the frog egg. And so their illustrations that accompany their paper in this French journal really provide a gorgeous illustration of the various stages in frog development, all the way from a fertilized egg all the way through the various divisions up to the embryo. And so their description is clearly the first really clear description of how the fertilized egg subdivides into parts as it becomes an embryo. But here again, this is 1824. This is before the cell theory really kicked into gear, and so they didn't appreciate either what they were looking at. They had no idea that they were seeing the formation of cells during each of these subdivision processes. And so it wasn't until maybe 10 years later or less that people started realizing, thinking about, cells became much more important at that point as a building block, and it was soon thereafter that people started describing what appeared to be cell reproduction in multicellular organisms. And so that was another big major event or landmark in the history. And the person who's generally regarded as the first person to witness cell division in a multicellular organism is this Belgian scientist by the name of Dumortier. This is an unusual copy because it's presented by the author Dumortier to a colleague. And so there's his signature. And he published this rather large article in 1832 in which he described a lot of different organisms, both animal and plant, in which he was just describing various aspects of their biology. And one minor little segment of the paper, he talks about this particular filamentous algae called silkweed or conferva, in which he was observing cell division at the very tip of the filament. And he provides a nice little illustration of that cell division process right here in this figure 15 of the first plate. So down there, what he says in the accompanying text is that that cell A at the very end of the tip elongates and then a wall forms right through the middle of it. And he then speculated that perhaps this is how cells are produced, by division, which was actually a fairly heretical notion at the time. And in later in the 1830s, when Schleiden and Schwann and all the rest of the big German famous cell biologists came along, most people did not believe in cell division. They thought that cells were reproduced by essentially being crystallized in the medium surrounding other cells, or perhaps they formed inside cells and then were then ejected. And so there was a lot of disagreement about how new cells formed, and this was the first clear evidence that they formed by division. And it was then a lot of other people started following up on this and seeing cell division in other organisms. Then eventually, of course, it took maybe 20 or 30 years before it became clear that all cells divide by cell division. And so by the end of the 1850s, essentially, a number of scientists were proclaiming that that must be the way it is for all cells. And then there's just one last little highlight I thought we should always mention, because anyone who knows anything about cell division, and oftentimes you see this guy in research talks because he is very famous for what he did, and that's a scientist by the name of Walter Fleming, who is essentially credited with sorting out how mitosis works and how duplicated chromosomes or sister chromatids are segregated on the mitotic spindle. And he has a ton of really outstanding work in the late 1800s. But his major discovery was, before he came along, people had seen chromosomes and they had seen these interesting behaviors that chromosomes do during mitosis. But to most people, these chromosomes looked just like blobs that were sort of nondescript. And then this big blob of chromosomes would separate into two blobs of chromosomes going to opposite ends of the cell. It was not clear at all, of course, at that point that chromosomes were the source of the heritable material, because genetics hadn't even been really discovered at that point. But people realized that those chromosomes must have something to do with inheritance. There must be some indication. But understanding how they separated was really unclear until Walter Fleming came along, and he appears to have been really technically skilled with various stains and fixation methods that allowed him to see duplicated chromosomes as individual threads. And in fact, the word mitosis comes from the word for thread, because he was the first to see chromosomes, sister chromatids, as separate chromatids. And that allowed him to realize that during mitosis, chromosomes are longitudinally split and move to opposite poles of the cell. And so it's not like some of them go in one direction and some go in the other. It's actually every chromosome is split down the middle and moves to opposite poles. And that, of course, really set the stage for genetics and inheritance a few years later. And so, as I said, he published an awful lot of different work on this subject. And I can't bring it all. I have a lot of it at home in various journals and so on. But I brought along the one that really established this idea of longitudinal division. It was published in this journal called the Archive for Microscopic Anatomy in Germany, which was published in these huge annual volumes. This is just one volume. They're all enormous. They're filled to the brim with beautiful illustrations of all sorts, which is of all sorts of different aspects of microscopic anatomy, all the way from tissues down to single cells and division and so on. And I actually, one of my big mistakes as a book collector is that I for some reason, bought 72 volumes of this journal. And so my dining room currently has a long bookshelf on it which has 70 versions of this thing, taking up an awful lot of space. So these books are filled with lots of nice illustrations, and this is where Walter Fleming published a lot of his major work. And what's fun about Fleming is that he was obviously very meticulous about his illustrations, as he had to be. To describe this longitudinal splitting, it was important to be able to really show clearly what was going on. And so his images and his illustrations tend to be among the most striking in the entire journal. And in fact, among the most striking of all cell biologists of the time. You can flip through a journal like this, and as soon as you come to the most gorgeous, beautiful illustrations in the book, you know you've found the Fleming. Okay, here's the illustration that established the idea of longitudinal splitting. Essentially, these two pictures right here show chromosomes not as individual rods, but as a series of a pair of lines that are longitudinally split. And you can see in a lot of other illustrations how he really emphasized this splitting of the chromatids into two. And so those illustrations really are the beginning of our modern understanding of mitosis. And that, of course, is why so many people love to use Fleming's illustrations as illustrations of mitotic principles. I would say that the interesting thing about learning about the history of a field like this is that it really helps us understand what we're doing today. A lot of students, especially young people, and I was the same way to some extent, don't really find history very interesting because they think it's all old stuff that's been sorted out. But I found by going through a lot of these old papers and going through the old work, I can see issues there that are exactly the same as what we deal with today. Issues like how to do a good experiment and what meticulous observation is worth and how important good description is as well, not just experiments, but good description, which a lot of people poo-poo these days, of course. But also issues like controversies between famous scientists in one camp and famous scientists in another, and how in the end sometimes the greatest scientists of a generation can actually be wrong, and that's always an interesting thing to observe, and there's plenty of examples of that in this history. But in the end, the important thing is to appreciate what a great experiment is, and even in the old days, Abraham Tremblay, sitting in this house in the countryside of Holland, was doing really beautiful experiments that were driven by pure creativity and curiosity. And we somehow have to capture that in the science we do today as well.